All right, so today I'll focus on some novel aspects of resource recovery, and these include, of course, production of bio-based chemicals, fuels, using biological platforms, and I'm going to specifically focus on the production of lipids in terms of the fuels, and methanol in terms of a representative bio-based chemical. And so this is not a talk just on energy. It's also a talk linking a few different sectors, including clean water and energy. And so before we go into this, perhaps we can take a look at how we address the challenge of providing clean water today. In the developed world, at least, we have clean water. That's a positive thing, but the way we get here is by designing and running massive systems to achieve this objective. And how do we get here? Let's perhaps take a very brief look at the process itself. So we have sewage, which we have to deal with, sewage, wastewater, and so on in the developed world, mind you. And essentially what we do is when we get the sewage in, we employ primarily physical and chemical processes to take out, let's say, the solids, the inerts, the inorganics for the most part. And then what we do is we channel what is remaining in terms of the carbon, the nitrogen, the phosphorus, the organics into fairly sizable biological reactors. And in these biological reactors, we engineer the processes of carbon redox cycling, nitrogen redox cycling, and phosphorus conversion. And in the process, what happens is that these contaminants, carbon, nitrogen, phosphorus, are converted into certain end products. Conventionally, these end products include carbon dioxide, nitrogen. Where there are nitrogen limits, we aim for the production of nitrogen gas, which is not a pollutant. It's just a constituent of air. And then phosphorus we just take out with the microbes which are produced. And then subsequently what happens is that the bacteria that we've grown in these biological reactors are just cycled back into the biological system, because after all, we've spent a lot of resources and energy in growing these organisms, so it makes sense to recycle them back. And then the treated wastewater could be disinfected, depends where we are, and then is discharged. Now let's take a look at just a deeper look into the biological system, which is where most of the biological activity is actually happening. And so what we are actually doing is, indeed, we are getting clean water from let's say sewage, but this comes at a cost, and what we are actually putting into the system is a lot of energy, and about 70 to 80% of this overall energy that goes into wastewater treatment goes into the biological system, and primarily that energy goes into the practice for providing air. We are talking about reactors, just to give a sense of scale. We are talking about bioreactors which are open for sure, but also which are about hundreds of millions of gallons depending where we are. So we are talking about massive systems, and to provide aeration to achieve a certain dissolved oxygen concentration in these reactors, a lot of energy is required. That's not all. These systems are also often typified by the consumption of a lot of chemicals to again achieve clean water, and these could run into several millions of dollars per year. And then in some cases what happens is that there could also be the production of certain greenhouse gases, including nitrous oxide, which is about 300 times as bad as CO2 on a 100-year lifespan, and methane, which is about 25 times as bad as CO2 on a 100-year lifespan. So these could also result from inadequately designed, operated, and monitored systems for achieving clean water. So the point is, indeed, we are achieving clean water, but this has come at some expense. Now, what happens is when we try to translate, when we just try to take this model and put this into other places, the model very often does not work. And it's not a question of money. Before we get into that, this is just a schematic of the low degrees of sanitation and wastewater treatment and sewage treatment that populations around the world, essentially, we don't. A large part of the world's population does not have access to the current models of wastewater treatment, current models of sewage treatment and wastewater, and so on. And so this is what we are dealing with, and this is just a snapshot of the western coast of Africa, where indeed this is Accra, by the way, and there is indeed a big wastewater treatment plant which is designed and implemented with assistance from the World Bank, but the plant does not get any wastewater or sewage or fecal sludge. And literally across the highway from this plant, you can see this plume of fecal sludge in the Atlantic Ocean. And this is one, I'm sure, of many examples where we've tried to implement forcefully, let's say, models that we are comfortable with for wastewater treatment and which are serving no purpose. So now, and the factor is not lack of finances. This project was completely financed externally and built, and this is a far more complex issue. One factor that leads to a picture like this is lack of energy required for achieving wastewater treatment, sewage treatment, and sanitation. It's not that communities don't have money. When we talk about energy, there are details that sometimes we don't focus on. In communities like this one, there is energy, but there's no provision of stable, reliable energy. People don't have access to energy 24/7. So when they do get energy, they are not going to spend it on treating wastewater. They are probably going to use it for other purposes, including lighting their households and so on. So these are the complexities that we need to address. And it's not energy as a whole. There are also dimensions associated with localized availability of energy. If there's no grid, how do we generate energy, perhaps within this plant, to keep this plant running? That becomes the question now. So then now the link perhaps begins to form. The question now becomes, is it possible to link other cycles, other sectors, including sanitation and human health, with the localized production of higher value commodities, biofuels, and chemicals, and not to compete with, let's say, refineries, but to keep the enterprise of sanitation running, for instance? This could be one link that we need to frame. This is another question that we need to ask and therefore answer. And so now, if we go back to our same system, perhaps the model looks somewhat different. So we could use the same infrastructure in place, or we could choose to change this, especially if there's nothing existing, as in some of these communities. So again, what can we recover, what can we do, what can we make, and where can we make this? These are the details that we need to look at. So if we look at just the raw fecal sludge, the raw sewage coming in, we could engineer technologies to recover biologically based chemicals and energy. In the reactor, surely, since these are almost biological factories, we could also focus on making chemicals and energy. And I'm going to leave energy a bit open, because there are different forms of energy that can be captured. This could include biogas, methane. This could include other end products as well. There are other examples of recovering flow energy from the effluent, just energy, because this is treated water after all. And then with some of the solid streams, these are very rich in terms of concentration, and they make near ideal substrates to recover, again, chemicals and different forms of energy. And now what we have done is we are not talking about wastewater treatment anymore. We are getting water, clean water, which is the bonus. Now this is one facet of water sustainability, energy sustainability, because we stop talking about this and we just look at a different question and answer a different question. And so in systems like this, what are we talking about? What does this even look like? So again, basically what we are talking about is carbon, nitrogen, and phosphorus coming in at let's say, different concentrations. And there's a certain hierarchy to this. First coming out of the picture is carbon. The reason is carbon, first of all, degradation of carbon is fairly is characterized by high kinetics. So the rates are fairly high, so we can put this right upstream, and so then carbon gets taken care of. Again, carbon is exciting because we can make a whole suite of chemicals from the carbon that comes in. Just to kind of open this up a little bit, the average oxidation state of carbon, organic carbon that comes in with all these different biogenic waste and food waste and agricultural waste is 0. So carbon is coming in at an oxidation state of 0. And imagine what we've been doing. We've been putting in a lot more energy, effort, resources to oxidizing it to CO2. Now what we are trying to do is change it, reduce the carbon in terms of oxidation state, perhaps go to biogas, which is -4, that's terminal reduction of carbon, and perhaps to other more exciting endpoints as well. Then what we are left with is phosphorus and nitrogen. Next in the sequence comes recovery of phosphorus where possible, where needed. Phosphorus is important to consider because we currently mine phosphorus, and phosphorus, we are going to run out of phosphorus at some point. We are not talking about completely offsetting the phosphorus demand using human waste, agricultural waste, animal waste, but these are offsets. These could be significant. Finally, nitrogen. In some economies where food security is of concern, we need to recover nitrogen as well. In developed economies today, there's no economic argument that can be made to recover nitrogen, and in this case, we have to look at other technologies to remove nitrogen in an energy efficient manner, and that's a whole other direction of research that my group is involved in. So this is the palette, and now we have to add on the dimensions of course, clean water being the endpoint, but also tying this to recovery of energy, chemicals, and so on, and making this whole enterprise economically viable. So the question is not, are we getting enough biofuel from this waste stream? That's not the question. The question is, are we making this overall activity economically viable? And that's even if we get there, that's enough. If more, then that's fine too. And so what can we recover? We can, based on technologies, based on biological processes, we can just about do anything. The question is, what should we be doing and where we should be doing this? And so without getting into those details for now, the options include biofuels, commercial chemicals, bioplastics, fertilizers, and many, many, many more options. Today, what I'm going to talk to you first is about the production of biodiesel, which is a fairly attractive endpoint in terms of being a biofuel, from fecal sludge. And fecal sludge is something that I was introduced to as a substrate stream about 5 or 6 years ago. Most of the world's population does not deal with wastewater. Wastewater is essentially human waste diluted by about a factor of 200. And the way I got this number is we can all calculate this. On average, we drink about 2 liters of water, half a gallon of water. And on average in the US, we produce 100 gallons per person per day of wastewater. So it's half a gallon consumed, produced 100 gallons of wastewater per day, per capita. And so this is where I get the 200-fold dilution from. And so, and this is just the way the infrastructure is laid out in the developed world. Most of the world's population does not deal with wastewater. They don't deal with piped sewerage. They deal with fecal sludge, which is mostly human fecal sludge, and this is basically conveyed to let's say treatment plants or dump sites just in an intermittent fashion, trucks for instance. And so what we wanted to do was we wanted to convert fecal sludge into a higher value endpoint, biodiesel. Okay, so let's take a look at what biodiesel is to begin with. So biodiesel is essentially the methyl ester of long chain fatty acids. So methyl ester, so we are combining methanol with the carboxylic acid functional group in long chain fatty acids. So this is biodiesel, and what happens as a result of the formation of biodiesel is that glycerol, which is the alcohol to begin with in lipids, gets kicked out, and methanol takes its place. So that's in a way that, in a simplified fashion, that's what is happening. We are taking the glycerol ester of long chain fatty acids, combining that with methanol. Glycerol gets kicked out, and methanol and the carboxylic acid functional group come together. That's biodiesel. Okay, so what do we need for biodiesel manufacture? If we can provide methanol externally, we need lipids. Those lipids are the substrates now. And so lipids are just long chain fatty acids, and this is the ester bond, glycerol, the 3-carbon glycerol, and then the fatty acid. So we are substituting the glycerol with methanol. Okay, and then the question is, if this is what we want to do, fecal sludge to biodiesel, if we are able to provide the methanol externally, the first question is, what is the lipid content of fecal sludge, right? So if we just want to go into fecal sludge and make biodiesel, then we need to know what the lipid content of fecal sludge is. Can all of you see the bottom panel here? Yeah, so basically this information is available sometimes with the medical records, but what we had to do basically was to run around these trucks that go around different communities taking fecal sludge and characterize not only the lipid content, but also all sorts of other parameters that fecal sludge can be quantified using. And basically what we find here is, just to kind of summarize this, fecal sludge in general contains about 6 to 9% dry weight lipids. That's what we're dealing with. And so if I want to successfully define in a few different ways, if I want to produce fecal biodiesel from fecal sludge and just focus on the inherent lipids, that's all I have to work with. The 6 to 9% for healthy individuals, that's what I have to work with. Now let's take a more detailed look at fecal sludge. So this is fecal sludge. Most of it is water. And what I'm mentioning here is that this is the tiny little fraction that we can directly convert to biodiesel by transesterifying with methanol. So this is the tiny little fraction that we have here. Of course, we can do this, and we've done this. We've done this not just in a tiny lab, in my lab. We've done this at pilot scale. So when I say pilot scale, this is the equivalent of 5,000 to 10,000 people per day in a pilot plant that we ran in Kumasi, Ghana for about a year and a half. Now, would it not be beneficial if we could focus not only on this tiny little fraction that we have, but if you could just develop a process that could convert all the organic matter in fecal sludge into biodiesel? It would obviously be far more beneficial than just focusing on that little fraction. Now, of course, it would be even more beneficial if you could develop a process that could take any organic stream, be it fecal sludge, food waste, animal waste, wastewater, sludge, and if we could make biodiesel from this. And that was really the question that we wanted to subsequently ask. And so that's what I'm going to describe today. And so the platform is this is the layout of the platform. So here we have basically just about any organic carbon. Any organic carbon except, don't ask me to convert a C1 compound because it's still quite different. But everything else is fair game. And so what we are essentially doing is anaerobically fermenting these carbon mixtures, dirty carbon mixtures, variable carbon mixtures, anaerobically fermenting them to produce short-chain volatile fatty acids, and which are then acting as the precursor to commodity chemicals, lipids, biodiesel, and so on. And that's where the benefit lies. And then what we have is we take the lipids out, make biodiesel. That's all fine. But then we obviously generate organic waste again. And what we now do is we channel all that waste into a much bigger anaerobic digester to produce methane and nutrients. So just to kind of explain this a little better, how is this different from anaerobic digestion? How is this more beneficial? Anaerobic digestion, as I'll show, I think, in a subsequent slide, is a very slow process and requires a much bigger reactor volume to achieve the same degree of conversion. In contrast, anaerobic fermentation, which does not go all the way to methane, does not require such a big system, requires a much smaller system. And so this is our overall approach. So just about any carbon into fermentation to produce lipids, and then the rest of the organics go into anaerobic digestion to still produce methane and nutrients. And depending where we are, methane can be used. This is not just methane, this is biogas. Biogas is about 60% methane on a good day. That's what we have. Biogas also has carbon dioxide and hydrogen and moisture as well. And so this is biogas. We can use this directly or use it or just flare it. And the nutrients, again, depending where we are, we could beneficially reuse the nutrients or figure out ways to treat the nutrients. And so let's take a look at anaerobic digestion. What are the different steps? Again, broadly speaking, anaerobic digestion consists of a cascade of steps going down from the hydrolysis of complex carbon molecules, basically 3 types of carbon molecules: lipids, the 6 to 7% dry weight, proteins, and carbohydrates, the sugars. And so that's what we have. And then these are hydrolyzed to their monomeric forms, so proteins to amino acids, carbohydrates to simpler sugars, and then lipids to long-chain fatty acids. And then the next process is acidogenesis, where these compounds are subsequently still broken down through, again, multiple steps to volatile fatty acids. These are short-chain molecules, about 6 carbons maximum, and then having a carboxylic acid group, COOH. So these are short-chain volatile fatty acids. And then finally, acetogenesis, which is the synthesis of acetic acid, which is a 2-carbon carboxylic acid. Now, this is where fermentation stops. And then from this step on, there's also the production of carbon dioxide and hydrogen. And the last step is really methanogenesis, which results in the formation of methane. The last step is the rate-limiting step here. The kinetics are far slower, and typically, for a temperature of about 35 degrees or so, we design digesters, anaerobic digesters, with a retention time, hydraulic retention time, which is basically describing how long the liquid stream stays in the reactor. So therefore, how big the reactor is. We design reactors of about 10 to 15 days hydraulic residence time. The HRT, the hydraulic residence time, is an indication of how big a reactor is for a given flow. Okay, so it can be used interchangeably if the flow is set. So this is the overall cascade of anaerobic digestion. Now, if we just look at fermentation, the first platform technology that I mentioned, which can basically take up any feedstock, we are only talking till the production of acetic acid. And here you see the hydraulic residence time, or the equivalent volume for a given flow rate, can be even as low as 2 days. So from a huge reactor, we have gone down to a much smaller reactor. And that's a benefit. And what we have now is not methane, but we have acetic acid, carbon dioxide, and hydrogen. And that's what we have. The big benefit of acetic acid is acetic acid can basically funnel into just about anything, into the production of lipids, into the production of other commodity chemicals, as long as we can take the acetic acid out of solution. And that we can do by elutriation. We don't need any energy-intensive or resource-intensive unit operations. And then finally, the advantage of thinking like this even is we don't have to design any new infrastructure. Where there are digesters, we can separate fermentation and digestion. If there are no digesters existing, we can build the system. And again, we are not saying this or that. We are saying fermentation as a precursor step to extract commodities, and then followed by digestion. We are not sacrificing one for the other. So this now becomes the overall platform. So what I'm going to, and so this is our process now, taking organic waste, and this is our system in Kumasi. Our system was 120,000 liters, and that was able to handle 5,000 to 10,000 people per day. And here we were producing volatile fatty acids. And these volatile fatty acids we were converting to lipids. That's the third step, and then lipids to biodiesel. So today I'm just going to focus on this third step, which is quite crucial. Of course, production of acetic acid is quite important, but I think we know a lot about production of acetic acid. This is really the novelty here. Okay, so for our study, conversion of volatile fatty acids to lipids, we wanted to test the impact of, basically, this is a process evaluation, which was done in the lab, both in the US and in Ghana. Basically, we wanted to look at the kinetics and the yields of lipid production from the fermentation of food waste, fermentation of fecal sludge, using synthetic volatile fatty acids, and then using glucose as a reference. Because, in a lot of studies, glucose is used, and the numbers are quite spectacular in terms of lipid accumulation, but fecal sludge is not glucose. And so this was the benchmark. And then we tested a range of initial volatile fatty acid concentrations, composition. Now, this composition of VFA, acetate, C2, propionate, C3, and butyrate, C4, this is what we got from fermentation of food waste, and that's what we used for subsequent studies. We want this process also to be quite flexible in handling different composition, different feedstock composition, and we impose different composition by manipulating the carbon to nitrogen ratio. So just to give you an example, if you're talking about fecal sludge, we are talking about a certain COD to nitrogen ratio, organic carbon to nitrogen. COD here is just a representation of the organic carbon content. So I'm just going to use that term instead of saying organic carbon over and over again. And so fecal sludge has a organic carbon to nitrogen to phosphorus ratio, which is quite similar to that of the dry content of wastewater. But then we did not want to be restricted to that. For instance, if we now talk brewery waste or or certain animal waste, the nitrogen content and the carbon content can be completely different. And we wanted this process to be able to handle all of this, and that's why we tested different feedstocks. The system that the biological system that we looked at was this yeast, Cryptococcus albidus. Now, essentially, what we're trying to do here is have Cryptococcus albidus convert volatile fatty acids, different composition, different concentrations, different nitrogen to carbon ratios into lipids. And we tested this using batch reactors in the lab or flow-through reactors in the lab. Now, this is just done in the lab here in the US and also in Ghana. So let's go to the results. So what I'm going to show you here is the other results in terms of concentrations of the cells produced, the biomass, grams per liter, and the lipid concentration, grams per liter, and the lipid accumulation inside the cell. So this is the lipid accumulations on the right-hand axis. The biomass and the lipid concentrations are on the left-hand axis. Now, this is plotted for different carbon to nitrogen ratios. The first point, COD to N ratio 5:1 represents excess nitrogen. And then we started for the same initial organic carbon fed, we started limiting the amount of nitrogen, so therefore the ratio goes up. And we wanted to take a look at the impact on these different variables that we measured. And basically, all we saw was that for going from a COD to N of 5:1 to 25:1, there was an increase in the internal lipid content. But thereafter, for all other ratios tested, the intracellular lipid content was close to about 28% as a dry cell weight. Now, this 28% is already among the highest ever reported for Cryptococcus albidus, not on glucose, but on volatile fatty acids as the organic carbon source. So this was just in a batch culture, and these results were quite promising. Now the question is, of course, we know that the system can handle a variety of carbon to nitrogen ratios in terms of lipid accumulation. But nitrogen is an essential nutrient. Are there any other responses? And to test this, what we looked at were not the yields, but now the kinetics. And so what I have here are these same numbers, the COD to N ratios, and then what the corresponding specific growth rates were. So now we are not talking stoichiometry, we are talking kinetics. And you see, for excess nitrogen, we have a maximum specific growth rate in the units of per hour. We have about 0.04 hours. With progressively increasing degrees of nitrogen limitation, the specific growth rate is compromised. So surely we are making more, we are kind of stabilizing in terms of lipids, but the specific growth rate is going down, which means now we'll need to build a bigger reactor, which we don't want. So for us, the 25:1 ratio was considered the most favorable based on this organism, based on batch cultivation. So clearly, we know that this process can handle variability in influent feedstock, but under the conditions that we ran the study, there seems to be an optimum value of carbon to nitrogen. Why is this information important? Let's say we are talking about variability in influent feedstocks or different feedstocks. We now know what, if needed, we can perhaps resort to blending feedstocks to arrive at a value which is close to what we get to be the optimum. That's important. We just can't be blind and just impose any carbon to nitrogen on these systems. Okay, now let's look at other types of yields and stoichiometry. So what I'm plotting here on the x-axis, again, different COD to nitrogen ratios, and then on the y-axis here is the biomass yield. So this is the amount of biomass, amount of cells of Cryptococcus albidus produced per COD, per volatile fatty acid organic carbon fed in. And we see, sorry, not fed in, amount of cells produced per amount of COD consumed, not fed but consumed, sorry. So this is delta X by delta COD. And we see that there's no change. There's no change. For the amount of carbon that these cells are cycling, they are producing the same biomass. But we have to be careful here. This biomass amount is measured gravimetrically. So if these cells are storing lipids, carbohydrates, or proteins, that is going to be reflected in this value of x. So it's not just cellular biomass, it's excess storage that has happened as well. So basically what is happening is that these cells are storing lipids as a possible storage compound.
Is the volatile fatty acid you're feeding pure acetic acid or a mixture of a whole bunch of?
Yeah. It's actually, these studies are done with synthetic acetate, propionate, butyrate in this specific ratio. And sorry, I should have mentioned that. And we chose this ratio because this is what we got from the food waste fermentation. So this was a good start. I'm not sure if I included the results from the actual food waste fermentate, but not in the results, but in the economic analysis I did. Okay, so basically what we are saying is that the cell mass, the total cell mass consisting of biomass and the storage compounds is basically the same, irrespective of how much nitrogen that they are consuming and carbon that they are consuming. On the other hand, what we are showing here is another type of yield coefficient, the delta X, so again, amount of cells produced per delta nitrogen consumed. Now, obviously, we are feeding it lower and lower amounts of nitrogen, but what we are seeing is that the cells are able to utilize nitrogen far more efficiently, the higher the degree of nitrogen limitation imposed, but then they are also able to store carbon in this, well, we measured lipids, but they are probably also storing other types of carbonaceous molecules, including possibly some sugars. So basically, this is what is manifested in terms of gross, in terms of bulk phase, bulk variables. Now, the question obviously is what is happening. So for this, we need to go into, let's say, the citric acid cycle for these lipids. And so the citric acid cycle consists of many, many different organic molecules, a lot of which feed into the energy synthesis pathways and the carbon skeleton pathways for biosynthesis. And in eukaryotes, of course, we are talking about these inside certain organelles. And so when everything is going well, let's say citrate is getting converted to molecules like isocitrate and then alpha-ketoglutarate. Alpha-ketoglutarate is a key molecule involved in nitrogen assimilation. And so when we are imposing nitrogen limitation, basically what happens is that isocitrate is transported outside the mitochondrion, and then isocitrate gets converted to acetyl-CoA, and then acetyl-CoA gets converted into intracellular carbon storage molecules. I'm sorry that that this is not showing up very well. So basically what is happening is that instead of going through carbon assimilation pathways, basically what is happening is that carbon, which is in excess now relative to nitrogen, is just being stored because the stoichiometric link is breaking down. Okay. So that's based on conceptual pathways that we know exist. This is not something that we have shown, but I'll lead you through certain pathways on how to even elucidate these pathways now, elucidate these steps now. Okay, let's come back to lipids for just a moment here. Here, of course, it's good to have a lot of lipids. It's, of course, good to have high rate of lipid synthesis, but it's also important to be sure that the right kind of lipids are being produced that can actually feed into biodiesel. And so here what I'm plotting are the results from different degrees of nitrogen limitation, also from food waste fermentation and from glucose. Without getting into each and every compound, if you just take a look at the fingerprint here, essentially these organisms are accumulating 3 types of lipids. And if we look at the composition, basically what we are producing under different conditions imposed, we are essentially producing the same lipids as we would produce from soybean oil or jatropha oil. And this is good because these are indeed the substrates which are used as feedstocks for commercial biodiesel production in the United States and the European Union. So we are indeed producing the right kind of compounds for feeding into the biodiesel pipeline. Okay, now even if we were to completely divorce ourselves from the links to clean water and sanitation and if we were just to do an economic analysis, this is how it looks. Now, the production cost of biodiesel depends on the chemicals that we are using, solvents, methanol, and so on, but also the source of the lipids themselves. And so here, we took the worst examples. So we took the the carbon source cost, which comes from fermentation of sewage sludge, which is not from our study, but this is $30 a ton if we are just talking about fermentation of sewage sludge. Fermentation to produce short-chain volatile fatty acids, $30 a ton. And then we'll come back to the red text in just a moment. And then we took the lowest yield from our Cryptococcus albidus experiments using anaerobic fermentate from food waste. Now, this is where the food waste BFA are coming. And so this was the lowest degree of accumulation that we observed, which we took into this economic analysis. And then if we go through the calculations, we are coming up with a biodiesel production cost of 71 cents per liter, which even if we were to completely ignore the other benefits, this is still coming out to be somewhat favorable relative to what biodiesel costs today to purchase, and that's fine. And so now let's go back to the red text. Now let's go back to let's consider what was happening in Ghana for just a moment. Fecal sludge, it's not collected. It's not fresh fecal sludge is rarely collected. Collection happens every day, but the collection happens from pit latrines, private septage, public toilets, typically containing aged fecal sludge, meaning the fermentation process has already started. We don't need the fermenters at all. The sludge is already coming in fermented. So this is gone for the most part. This is completely gone. And so now this is why I wanted to point out the singularities of fecal sludge as well for just a moment. Fecal sludge is not wastewater. Fecal sludge is not sewage sludge. Fecal sludge is completely different. So the way we don't have to design a 20-day or a 15-day or a 2-day retention time reactor even. We just take it and directly go to chemical synthesis because it's already there fermenting. It's probably there also producing methane by itself, but that's not a good thing. But at least the VFA are there. All we have to do is collect the VFA, channel them to chemical synthesis, and go towards digestion. That's what we need to do. And so this is gone. Perhaps a tiny fraction remains, but we don't need a reactor system here. We just take the fecal sludge and go forward. And so then this starts to become even more favorable. And right here, we are not even taking into account the positive impacts of, in terms of human health and environmental health, which can be monetized. We are not taking that into account in order to come up with this figure. And again, the point I would like to make here is we are not competing with the biodiesel industry. What we are doing is, all we are trying to do is we are trying to render the efforts for improved human health, improved water quality, and improved sanitation. All we are trying to do is make that possible even. Because today the biggest barrier against some of these efforts is lack of energy, local available energy. So for us, we were sitting on a dump site in Kumasi, Ghana, which is, there's nothing else there. There's just fecal sludge, there's a landfill, and there are trucks coming in every minute. There's nothing else there. There's no energy. So if we can make enough energy to offset or make this process energy neutral, then this is quite good, I must say. Okay, so there's even more excitement lurking in this overall study, and I'm happy to report some of this. Cryptococcus albidus is we find to be a good medium to channel VFA, volatile fatty acids, to lipids. We wanted to go into those pathways that I conceptually laid out. This is based on what we know from biochemistry and biology textbooks. Now we want to actually see what is going on, and to do this we need let's say we need some information in terms of the genome itself, and we found that the genome was not available. And so in the last couple of months, what we've done is we've gone ahead and sequenced the genome of this organism. The average length of the genome in Cryptococcus albidus is about 25 megs, and basically what we are doing is through our sequencing efforts, we are sequencing the genome, just to put this in general terms, 30 times. So we're getting rich information. So we're just confident in our prediction of the genome sequence. And for this, we are using another Cryptococcus strain as the reference. And so what we are getting is information like this. This is at the DNA level. The genome essentially gives us an indication of the potential of what an organism can do. At any given time, just a tiny fraction of this potential is being expressed, 25 to 40%. Depends on the environmental conditions as well. But this is what we are getting. When we get the sequences, we are classifying them under different categories: metabolism, cell processes, signaling, information storage, processing. And given where we are today in our knowledge of microbial systems, you will always come up with sequences that we can't interpret, and we lump them into poorly characterized or uncharacterized. This is what the genome looks like, and these are the number of sequences that are allocated to each category within the genome. And now what do we do with this information? We have the entire, so this is, let's say, the complement of different metabolic pathways. So basically what we have are, let's say, biosynthesis of metabolites, metabolism of cofactors, different categories: lipid metabolism, which is what we are interested in, the TCA cycle, which is what we are interested in, nitrogen metabolism, which is what we are interested in. We have the whole metabolic map laid out. The way to read this is as follows. Let's say these are different reactions. Anything that shows up in blue basically means that this genome has that sequence or a related sequence. Now we have this information. Now we can go back to our lab, run the Cryptococcus cultures under different conditions. Since we have the genome, we can figure out which pathways are turning on, turning off, to what degree. It's not 0, 1, it's also to what degree, and we can do this quantitatively. We can do this at the messenger RNA level. If you want to look at gene expression, we can do the proteome as well if you want to look at protein concentration. So now we have this, information to use. And so for the first part of this talk, so what we have done is we have basically come up with a microbial platform, a yeast-based platform, and we've also worked with a fungal strain, Fusarium, which I did not present today. We have a platform now to basically go from volatile fatty acids to lipids. We know that we can produce volatile fatty acids from just about anything at very short hydraulic retention times, and that's really the attraction of this fairly flexible platform. And biodiesel lipids are just one endpoint that we can achieve using volatile fatty acids. Volatile fatty acids really open up the door to many, many more endpoints. And for us, for now, I'm happy to say that we are using this to link sanitation practice, environmental health, human health with energy and chemical recovery. So that's the first part of the talk. Should we have questions now or just go through it and then have questions at the end? Keep going. All right. So now the second portion of the talk is, let's keep going downstream in a sense. So let's take a look at not only VFA to biodiesel. Now let's take a look at anaerobic digestion and see what we can do with biogas. And so this is where we stopped last time. So with biogas and nitrogen and phosphorus. Now people have been producing biogas for centuries, and we don't know how to do it yet properly, especially when we start talking about fecal sludge, which is already pre-fermented, pre-converted. We just don't do this correctly. And biogas, of which methane is a component, it's not trivial to use biogas. Even in the developed world, if you look at wastewater treatment plants around the country in the US, surely they produce biogas. But most plants flare the biogas. And the reason is because they don't produce enough biogas, and they don't produce enough good quality biogas. So they just flare it. And so the question here is, can we do something? Can we do something with this? And so what we have done is we have developed another process, another technology, which can take the biogas, the methane in the biogas, and we're not worried about the carbon dioxide, the hydrogen, the moisture present in biogas. And this bioprocess technology can take the biogas to methanol, the methane in biogas to methanol. And the other benefit is that it is actually driven by the nitrogen, which is coming out from the bottom of the digester. In many cases, what happens is there are many discussions about converting organics to methane. Now, anaerobic digestion does that quite beautifully. What anaerobic digestion also does very well is that it separates the carbon, organic carbon, from the nitrogen and phosphorus. So surely we can make a lot of energy from biogas, but then we need to know what to do with the nutrients. Nutrients cannot just be discharged into the receiving water bodies. Now we have a process that can take both these streams and convert this to methanol. And then again, we don't have to worry about the water quality impacts of the nutrients, the air quality impacts of methane. And then water quality, environmental health is the bonus. And we are also getting methanol in return for this. And so this was the basis of a previous publication, and then some of the more mechanistic work is actually I have some results to present as well. So first of all, why methanol? Methanol is a single carbon alcohol, CH3OH. Methanol is again used as a building block for if you look at commercial commodity chemicals, methanol is there. Methanol is also used as in its dimeric form as dimethyl ether, and dimethyl ether is also quite widely used as a fuel additive. In many parts of the world, ethanol is used as a gasoline additive. In China, methanol is widely used instead of ethanol as a gasoline additive. And then finally, methanol remains to this day one of the most widely used chemicals for nitrogen removal in the framework of biological wastewater treatment. To this day, methanol is the most widely used chemical. And so the applications are there. How do we get methanol today? Most methanol is still produced from the mobile process, which is chemical catalysis. The substrates are fossil carbon, wood, sludge, and so on. It's a chemical process, highly energy redundant, and the chemical process cannot deal with moisture and CO2, which is produced in, which is present in biogas. So these constituents need to come out. That's how we get methanol today. There's also potentially a huge source of methanol that we are faced with when we start talking about, well, tapping into some large reserves of stored natural gas as well. So now we are talking about liquid streams resulting from fracking operations, which are rich in methanol, if not saturated in, not methanol, methane, which are very rich in methane, and these are now being viewed negatively as a waste, which wastewater treatment plants will not take. And the point is we have these methane-rich streams which can be better used, basically. And so how do we biologically convert methane to, how do we oxidize methane? And so we can, of course, rely upon methanotrophic organisms, which are methane oxidizing organisms. They like methane, and this is what they do for a living. They oxidize methane to methanol by the action of this enzyme methane monooxygenase. But the point is they just go all the way to CO2 because this is how they get energy. These are the methanotrophic organisms. And there are two distinct types of enzymes involved, the particulate methane monooxygenase and the soluble methane monooxygenase. So yes, we can produce methanol, but commercially from a process perspective, there's the prospect of running all the way to CO2, so it's not very favorable. What we did was we realized, and again, based on looking at many different genome sequences and looking at older literature, that there are other organisms which can go from methane to methanol, but predominantly stop there. And these are not the methanotrophic organisms. These are the ammonia-oxidizing bacteria. Now, what do ammonia-oxidizing bacteria do for a living? They oxidize ammonia. But instead of methane monooxygenase, they have a membrane-bound ammonia monooxygenase. Now, ammonia monooxygenase and particulate methane monooxygenase have evolved from a common ancestor, and it's just fascinating that they still have retained that function of being able to oxidize each other's substrates. So ammonia monooxygenase can oxidize methane. Methane monooxygenase can also oxidize ammonia. So this function is retained, and based on our evaluation of genome sequences of ammonia-oxidizing bacteria, they cannot produce CO2. They just don't have that step, and they predominantly stop at methanol. So conceptually, again, the approach is the same. Conceptually, we find something, can we harness this potential? And so this is the electron-based pathway for ammonia oxidation. So ammonia to hydroxylamine is a 2-electron transfer catalyzed by this monooxygenase, and then to nitrite, which is a 4-electron transfer. The oxidation of hydroxylamine and ammonia are coupled because of the 4 electrons which are released, 2 go back through intracellular electron pools. This is ubiquinone, but there are other electron pools as well. 2 electrons go back to this ammonia monooxygenase. A classical monooxygenase requires molecular oxygen, the substrate, in this case ammonia or methane, and reducing power. So this is very important to keep in mind, the reducing power, and that's how the system works. The analogous system in methane is, of course, methane to methanol to formaldehyde to formic acid to CO2. And again, it's a very similar model if you take a look at these compounds. And there's been a lot of work done by other groups on these systems, and also from all of the remaining are from our group as well. In terms of metabolic modeling, in terms of understanding gene expression, there's some amount of work that exists. And so this is our conceptual system. So we have a nitrifying reactor consisting of ammonia oxidizing bacteria. We have the digester producing biogas and ammonia. We feed this to the same reactor, and we have the ammonia oxidizing bacteria oxidize ammonia to nitrite, and then methane to methanol. And in this reactor now, what do we have? We have nitrite and methanol. What do we do with this? We switch off the air, and then we have denitrification take over to convert the nitrite to nitrogen gas using methanol as the carbon source. And this is, you know what we have done here? We have stopped the plants from flaring the methane and buying methanol. We are disrupting that link. We are taking methane to methanol and removing the nitrogen. And so this is what we have achieved here. And again, if you look at these organisms, CO2, ammonia oxidizing bacteria fix CO2 to make biomass. They actually like the CO2, and moisture is really not an issue. And so this is a very nice system that allows us to combine the microbial nitrogen and the carbon cycles, and this was the basis for the 2010 Paul Bush Award. And so again, so we've gone into this system. Ultimately, what we are trying to do is develop a technology and optimize conditions for oxidizing methane to methanol, and then really to maximize the kinetics, maximize the yields. That's the ultimate goal. Of course, there are a lot of steps. These are the first experiments that we did. Very excited with this concept. Went into the lab, did some experiments. Look at these concentrations, 0. There's no methanol. And so, of course, well, there was only one direction to keep moving in, that was forward. So we kept moving forward. We tried different combinations. And basically, in other experiments where we continuously provided methane and oxygen together to our ammonia oxidizing bacteria, we did produce methanol. And then of course, at this point, it was good. So what we then did was we went back to the schematic and realized that we were providing ammonia, we were providing oxygen, we were providing methane. What was missing was we were not providing enough electrons or reducing power. And reducing power strictly is only provided by hydroxylamine oxidation. And this is where the cells get their energy, not from oxidation of ammonia. And so what we did was we went back and looked at ways to provide electrons. And the first step was to provide not electrons, but hydroxylamine. And when we started doing that, things started to look better. We also had to resolve, and that's how we resolved the issue of reducing power by providing hydroxylamine first. That was the first step. But then we also had to overcome this impact of competitive inhibition. Now, what is this? We have ammonia monooxygenase that typically oxidizes ammonia. But then it can also oxidize methane if it is in the system. And when ammonia and methane are both present in the environment, they will compete for similar or same binding sites on ammonia monooxygenase. Now, we have to recognize and we have to quantify this and overcome this inhibition by engineering the reactor properly. But before we engineer the box, we have to look into the system and figure out what is really going on, and or play with the feeding strategy. Without making this too confusing, I'll basically tell you what we did. This is not showing up clearly at all. There's supposed to be a gray rectangle around this. So this should be gray, white, gray, white, gray, white. And the gray rectangles essentially describe where we are providing oxygen and ammonia. So this is how we keep the pump primed. We don't want the ammonia oxidizing bacteria to fall asleep, literally fall asleep, by turning down the expression. So we provide ammonia and oxygen. So the expression of AMO increases. Then we have the methane. We shut off the oxygen, and we allow methane to be oxidized. So now what we are doing is we are avoiding competition between ammonia oxidation and methane, and we are also providing reducing equivalents. So that's how we were able to overcome this. And we had to quantify how much nitrogen we need to put into the system based on theoretical limits, theoretical calculations of maintenance energy, and so on and so forth. And once we did that, we were able to generate far higher quantities of methanol. This is all in a batch system, so over time. So what I'm plotting here are the methanol concentrations produced, again in terms of the COD content, and over time. And so the different treatments here are as follows. Just putting in ammonia, which is this, and this gives us the lowest amount of methanol, and it's also leveling off. And then we have together hydroxylamine by itself, and then putting in together hydroxylamine and ammonia. That gives us a slight improvement, not by much. And then we have alternating ammonia and hydroxylamine and methane separately. But then finally what we did was we did something different. We took the biomass out, washed it, put it back in the system. These are still batch experiments. And I'll tell you, even though this looks a bit confusing, the reason why we did this was as follows. We suspected that the methanol produced was inhibiting in a feedback manner the ammonia oxidizing bacteria, and we also wanted to put more reducing equivalents. So when we started washing the biomass, the tapering off of methanol concentrations was even less pronounced. And here, these concentrations, these rates that we achieved, these are without any comparison. Here, this is what we have here. This is the. Now let's just focus on this profile here. These rates and the concentrations that we get are the highest ever reported with ammonia oxidizing bacteria and methane, for methane to methanol. And this was the proof of concept that we showed, and quite favorably in terms of both the kinetics of methanol production, in terms of milligrams methanol COD produced per milligram cell COD present per day, and the peak methanol concentration. Now, these numbers, let's try and put these in perspective. 60 milligrams COD of methanol can remove about 15 milligrams nitrogen per liter. 15 milligrams nitrogen per liter is the difference between a plant meeting permit and a plant not meeting permit. The penalty for a plant not meeting permit is about $35,000 a day. The other option is to buy $15 million worth of methanol per year for 5 plants out of 14 in New York City. So that's the difference. So this is what we are doing. And we are doing this in a retention time of 6 to 7 hours. We chose 7 hours because that's the average retention time of wastewater treatment systems in general in North America at least. So again, this was the proof of concept that we developed. We know we can produce methanol. The rates are good. The yields are good. The next step was to do continuous flow experiments because we don't want to be running batch systems. And so what we did was we have a system in the lab that treats synthetic digester effluent or synthetic urine, for instance. We were taking the biomass from here, and we came up with a different reactor design which simulates a continuous flow environment in a plant. And these are the results that I'm going to run through this just a little bit here. So this is done in the lab. This is the biomass concentration. This is the liquid flow rate. And there's a certain ratio of air to methane. The nitrogen feed is hydroxylamine first, then switch to ammonia, and everything is controlled. And so the summary here is as follows. When we put hydroxylamine into the system, we get a lot more methanol than when we just put ammonia into the system. Now, why hydroxylamine? Because this is the source of electrons. Ammonia, of course, but then electrons are just getting channeled all over the place, and methanol yields are lower. So the point here is, yes, indeed, even in a continuous system with continuous exposure to methanol, to methanol and methane, we are able to produce methanol. And the point is, hydroxylamine gives us much higher yields and rates, but hydroxylamine comes at a price. Ammonia is free. In fact, ammonia is a pollutant, which we are able to address in addition to the methane. Okay, so these are all process level studies. We are also opening up the system in terms of what happens to ammonia oxidizing bacteria when they are subjected to methane instead of ammonia. What happens inside the cell? How are the metabolic pathways being regulated? And so here we are basically running pure culture chemostats. Now we've gone from the field to the lab, mixed culture, to pure culture chemostats, where we run the chemostats to steady state, exposed to methane, go back to ammonia. So ammonia, methane, ammonia, and we are taking measurements of gene expression, protein expression, and so on and so forth. These are the results. So basically what we are doing is we are feeding ammonia to the system. The cultures are removing all the ammonia. They are producing nearly in stoichiometric quantities nitrite. So the system is doing well. And then for a certain period of time, we stop the ammonia feed no, sorry, we don't stop the ammonia feed. We just put in methane into the system. And then what happens as a result is now ammonia and methane are competing. And so as a consequence, ammonia starts to build up, nitrite starts to come down, and at the same time, methanol starts to build up. Methanol starts to build up, and these concentrations are, again, fairly high. And now we are taking samples before methane, during methane, and after methane exposure. And now what we are trying to do is, again, I'm going to present just two profiles. Now we have the genome of this organism available, so we can look to see what is happening. We can look to map out the changes. And so this is just the nitrogen metabolic pathway in Nitrosomonas europaea. The green symbols mean that these proteins exist in Nitrosomonas europaea in the genome. The red rectangle around them basically means that we detected these in our proteome. Let me not spend too much time, if it's okay. What I'm going to do is summarize this by saying, basically, there's hardly any difference in terms of the expression of the proteins, except that they are turning down. But for ammonia assimilation, which is basically going up. Basically, what the cells are doing is when they are flooded with carbon, a non-preferential source of carbon, they are just turning up their ammonia assimilation systems just to be sure that they can survive. This is on the assimilation side. Of course, the carbon cycle is far more complex. And basically, what we are seeing is that there is a net reduction in terms of ammonia oxidation, but in terms of production of acetyl-CoA, which can lead to carbon storage, the cells are basically turning up some of these pathways. But this is something that we really have to dig into. These are results that we just got last week, so I'm sorry that this is somewhat premature, but now we have the entire proteome to describe methane metabolism in nitrogen cycle organisms. How do we use this information? We can use this information not only in terms of reactor models, but we can go into the systems and look at metabolic models. This is one of the first metabolic models for Nitrosomonas europaea, this organism that we use in our methane to methanol studies. One of the first metabolic models that was published for this organism using a flux-balanced approach, and that's how we can use this information. And so finally, so in summary, at least for this section, we have developed a proof of concept to convert biogas into methanol, and now we are actually going into more details in terms of looking into the system, and ultimately we are looking to also scale up this process, again, for mixed cultures, not for the pure cultures. And so finally, as a vision, we've talked about recovery of resources, different endpoints, different technologies, perhaps different local considerations as well. Of course, we are quite excited by this. What my group does, basically, we would like to address, and we are in the process of addressing some of these challenges. Our approaches are more, first of all, understanding the system. Understanding can be at the process level, at the microorganism level, and much further down at the omics level as well, DNA, RNA, protein, and so on. Not just pure cultures, but communities of organisms, consortia of organisms, which make these systems more robust. We are interested, and we are doing a lot of technology development built upon these foundations in terms of resource recovery. And then some of these have been translated to scale in both the developed world and developing worlds, and this is something that we see being quite active in. Ultimately, and we are also closing the gap on not only water, energy, nutrients, but also food. This is from the Sustainable Food Project at Columbia University. Today, we don't associate Manhattan with agriculture. Might have been 150 years ago. But now what we are doing is we are converting food waste in our buildings, in the university buildings, into different endpoints, including lipids, including methanol, and nutrients for urban agriculture. And this is something this is a vision that we are truly excited by. And as you can see, we are not focused along a certain axis. We are really focused along a much broader nexus. Thank you. Sorry for all of this information, but if there is time, I can take questions.
So the growth rate of that ammonium oxidizer with using methanol versus ammonium, is it the same, the carbon fixation?
The specific growth rate on methane is much lower because what happens in methane to methanol oxidation is that the reducing equivalents are not produced. Because for ammonia to hydroxylamine to nitrite, it's really the reducing equivalents which keep the AMO primed. Without the reducing equivalents, there is no ammonia oxidation. AMO is not active. So this is very similar to the old co-metabolic literature where we had to provide external equivalents. And so if you don't have reducing equivalents, how is CO2 fixed? Using electrons. Those electrons are not coming into the picture at all, so everything suffers. Anabolism, catabolism, everything suffers from a process perspective. But inside the cell, obviously, changes are taking place, and certain pathways are getting overexpressed just to prepare, let's say, for starvation and so on and so forth. But at the process level, the rates are getting compromised.
Hi, thank you. I'm just wondering about your choice for the organism for your biodiesel system, Cryptococcus albidus. I was wondering, why did you select this particular yeast? Why didn't you consider, for example, other yeasts that are more sensitive to the nitrogen level? And I guess I have two questions. That would be one. And the other one is, given the variability of nitrogen that you would expect to get in your feedstock, would you contemplate, say, for example, combining the second system you described to reduce ammonia and then try to regulate the nitrogen content for your second step? Yeah, so the first question is why we picked this organism. There were a couple of reasons. Based on, again, there was some information in terms of VFA conversion to lipids, and the intracellular accumulation reported was somewhat favorable, and that was one reason for us to go with at least Cryptococcus albidus. The other reason was in terms of why we didn't perhaps pick algae. It's very difficult to get algae to make lipids from VFA, and I don't think you're asking that. Yeah, that reason was fairly straightforward. We just looked for systems where intracellular accumulation was reportedly quite high, and then we went with that. Another feature of this particular organism is that the temperature optima are around 27, 28 degrees Celsius, and that's where some of these streams come out. We don't have to necessarily heat up the wastewater to get higher rates, and so this worked out almost perfectly. And for us at that point, these were two good reasons for us to stick to this organism. The second question was, would we combine integrating some streams? The answer is absolutely yes. Depends. Of course, then what we need to do is we need to monitor what comes in, the carbon to nitrogen, which can fluctuate also. And then it's just a matter of equalizing the carbon and the nitrogen. If we have a carbon-rich stream, yes, we can use it to equalize it. Or if you have a nitrogen stream, again, we would either dilute it or concentrate it. And that's the way to maintain a certain ratio. But the point is, it's not one single number. This organism is okay. It can tolerate slightly different or variabilities in carbon to nitrogen without taking too much of a hit in terms of the maximum specific growth rate until it gets to really limiting conditions.
Could you say again what distinguishes or what conditions distinguish the digestion from the fermentation?
Yeah. And so basically, broadly speaking, anaerobic, so the reaction that we are missing out when we consider just acetogenesis, production of acetic acid compared to methane, is that last conversion of acetic acid to methane, or carbon dioxide and hydrogen to methane. So that's the principal difference. The main outputs from just fermentation are a mixture of short-chain volatile fatty acids, whereas from digestion, the main carbon compound really is methane. That's the main. I'm just looking for this.
How are the conditions?
Oh, sorry. so in terms of conditions, anaerobic digestion is most favored at, again, favored by low variability in the influent feedstock. pH ranges more in the physiological optimum range around 7 or so, whereas with fermentation to acetic acid, this process is, the kinetics are much higher, and it can also tolerate lower pH values. And temperature, I think we've run across a whole range of temperature, and I think if the reactor is designed adequately, the system can take temperature changes. But the last step is the slowest one. That's the one to keep in. That's important to keep in mind. Conversion of short chain fatty acids like acetate, CO2 and H2 to methane. That's the slowest step. And the other obvious big difference is that this step is catalyzed by completely different organisms. They are not even bacteria. These are archaea.
Are there any further questions?
Thank you very much. So, I mean, in the beginning, you showed a plant that can't operate because it doesn't have energy. So, presumably, your systems are close to operating as closed systems that produce enough energy to run themselves. How close are you to that? Is there excess energy or just a little bit short?
So the answer to this depends on what the endpoint is. So if you're talking about methane, all we need to do is import more and more carbon, make a lot of methane, and clean it and feed it to the grid. And so this can be done. Now we have to, if energy is the only option, we can do it this way. But there will be negative impacts on the environment because of the nutrients getting discharged now. So we have to take that into account as well. And again, that's why I put one of those earlier figures there, the flow sheet, because then we have to think not about nitrogen recovery and phosphorus recovery, but energy efficient ways to remove the nitrogen. Because we'd of course like to make the system energy positive, energy neutral, but that can't come at the expense of nitrogen and phosphorus. So it depends what we are trying to do. And these are examples of energy positive systems. Our system in Ghana was not even energy neutral, but that's the direction that we're trying to go in. And biodiesel helps because it's much higher on the chain than biogas.
Okay, let's thank Dr. Chandran for a stimulating talk.